Welcome to CTD130, Lifespan Development. In this lecture, we will cover the biological and evolutionary theories of human development. By the end of this lecture, you'll be able to describe the structure and functions of genes, describe the ways genes influence development, describe how epigenetic mechanisms regulate genes and development, describe how evolutionary theories explain individual differences, and describe how biological and evolutionary theories contribute to our understanding of disease processes and interventions. Understanding biological processes is crucial for grasping evolutionary theories of development. Genetic and epigenetic factors, alongside environmental influences, shape our health and well-being throughout life. Epigenetics offers new insight into the interplay between nature and nurture, potentially transforming our views of human development. Evolutionary theories propose that the genetic and physiological processes that underlie human development and behavior change gradually over time through genetic mutation and natural selection. Evolution is used to explain cognitive and, by extension, social and cultural behavior. Our body, cell's nuclei, contains 23 pairs of chromosomes that are made up of an astounding almost 2 meters of finely coiled DNA molecules. Each chromosome contains segments called genes, each of which influences a particular trait or developmental pattern. The DNA is a molecule that contains the instructions for how an organism develops and functions. An essential function of genes is to instruct body cells to combine 20 standard amino acids to build the proteins that our bodies need to function properly. Proteins are organic compounds that form the structures of the body and regulate its maintenance. Basic structural elements of our bodies, for example, muscle, brain, and bone, are made up of different proteins. A genome is all the DNA an organism possesses. The genotype is the actual DNA material that determines each person's unique genetic blueprint. The phenotype is the individual's whole set of observable characteristics. One way to remember the distinction between genotype and phenotype is that the phenotype can be identified by directly observing the individual. For example, you can easily see that a woman has brown eyes, which are part of her phenotype. Her genotype, though, can't be so easily determined. The simplest genetic rule is the dominant recessive pattern, in which a single dominant gene strongly influences the phenotype. If a child receives a single dominant gene for a trait from one parent, the child's phenotype will include the trait determined by that gene. In contrast, a child's phenotype will include a recessive trait only if she inherits a recessive gene from both parents. Human geneticists have learned that both dominant and recessive genes differ in expressivity, meaning that the degree to which any gene's influences phenotype varies from person to person. For example, all individuals who have the gene for curly hair don't have equally curly hair. So, even when a child receives the dominant gene from curly hair from her father, the amount and type of curl in her hair probably won't be exactly the same as his. Polygenic inheritance involves multiple genes influencing a phenotype. For traits like skin color, geneticists believe children inherit 3 genes from each parent. Dark skin is dominant over light skin, leading to a blend in offspring. If one parent is dark-skinned and the other is fair-skinned, the child will have a skin tone between the two, darker than the fair parent, but not as dark as the dark-skinned parent. Many physical traits are influenced by both genes and environment, a pattern known as multifactorial inheritance. Height is one example. Many genes contribute to a child's height and rate of growth. Scientists have discovered some additional principles of genetic inheritance involving genetic material that is found in the mitochondria rather than in the nucleus of a woman's eggs. In mitochondrial inheritance, children inherit genes that are carried in structures called mitochondria, which are found in the fluid that surrounds the nucleus of the ovum before it's fertilized. Consequently, mitochondrial genes are passed only from mother to child. And finally, epigenetics is the study of how our environment influences our genes by changing the chemicals attached to them. What we eat, our physical activity level, access to resources, and more affect those chemicals, in turn shaping our health. Epigenetics can help scientists understand why diseases happen and explore new avenues for treatment. Evolutionary theories attempt to explain our differences as individuals and our commonalities as a species. These theories often focus on the genetic and environmental mechanisms that underlie development throughout the lifespan and across generations. Ethology emphasizes genetically determined survival behaviors that are assumed to have evolved through natural selection. For example, nests are necessary for the survival of young birds. Therefore, ethologists say evolution has equipped birds with nest-building genes. Behavior genetics explores how heredity influences individual differences. It suggests that traits or behaviors are more similar in related individuals, like children and parents, than in unrelated people. Researchers have demonstrated that heredity impacts various traits and behaviors, including intelligence, altruism, substance use, and hyperactivity. Evolutionary psychology is a unifying approach to explain human behavior that involves all branches of psychology and life sciences. In particular, evolutionary psychology looks at how genetically inherited cognitive and social traits have evolved through natural selection. Evolutionary theory also and the research also are making science more aware of the relative importance of early life events on altering susceptibility to chronic, non-communicable diseases, as well as socioeconomic disadvantages in later life. This growing awareness has focused attention on the need to promote early intervention strategies not only during prenatal development but also before conception. For example, supporting good health and nutrition and social security in females of reproductive age versus interventions instituted later in life. This early intervention has the potential to prevent or at least minimize chronic diseases and social disadvantage in future generations. In summary, biological theories focus on the contribution of hereditary genetic mechanisms in development. Geneticists distinguish between the genotype, the pattern of inherited genes, and the phenotype, the individual's observable characteristics. Thirdly, inherited and acquired epigenetic mechanisms regulate gene expression. Fourthly, evolutionary theories take the view that through a process of biological evolution, the body, mind, and social behaviors have been shaped by natural selection to serve adaptive functions and promote survival. And lastly, after conception, both external and personal factors act to modify epigenetic regulators. These factors can alter patterns of gene expression and suppression and play a role in disease processes. Epigenetic changes can influence the health of future generations. The reference for this lecture is Lifespan Development, pages 28 to 38 by Boyd, Johnson, and B.